Sir, you can start, sir.
Good evening, my dear students. You all are welcome to the another session of the biological class. So last class, what you have seen already, about what is by immunity, what are the technical terms, and then antigen, antibody, agglutinin. Okay. So that things already we discussed, and what are the types of immunity, and innate and acute, and also divided into two again, passive and innate immunity, isn't it? Again, they divide into according to their natural as well as the artificial, like that. They may be divided. Already we saw it, that things also. And then, what are the types of cell present in the body? That things also already we discussed. What are the cell present in it? Already about. There are 5 types of cells have been reported. So what are the name? First one is called as helper cells. And another one is called as killer cells. And third one is called as memory cells. And fourth one is called as suppressor cells. And then last one is called as antigen presenting cells. These are the cells already we discussed. And then we will pass on to the next one is called as, which one antibody. The antibody collectively called as, what is that? Immunoglobulin, because globulin protein molecule, they have the ability to fight against many diseases. That's why they called us, what is that? Immunoglobulin, protein molecules. Before that, what are the things? So aims, what is the meaning of aims? Already we discussed cell-mediated immunity. That things also already discussed in each and one by one. Okay. And the last one is already we discussed, the antigen presenting cells. Antigen presenting cell means the each and every cell, they are outermost cell wall, they are having a, which one antigens. That's why they are called as antigen presenting cells. Okay. Next, AMIS, antibody mediated immunosystem, already we discussed. How many percentage present these B lymphocytes secrete the antibodies? How many percentage of antibody secreted by the, which one B lymphocyte by humoral immunity? So that things also already the last class we have discussed, isn't it? Say antibody. Today we are going to discuss about deeply what is my antibody, where there is a secretions. So it also comes under the heading is known as which type of defense is, it may becomes under the headings third line of defense, isn't it? Like that it comes. See that one by one. Antibody or immunoglobulin. Globulin is nothing but a protein molecule. It has the ability to fight against many diseases. That's why they are collectively called as immunoglobulin. So the immunoglobulin is also called as antibody. Antibody is also called as immunoglobulin. See here, the antibodies are third line of defense. They may ask the questions NEET. First line, second line, third line. Third line of defense is immunoglobulin, otherwise called as antibodies. They are proteinous substances present in the blood plasma. Already we discussed plasma. We are having the blood, isn't it? That blood contain 2 parts. One is called as, what is that? Solid part, and another one is called as liquid part. The solid part is 45%, and the liquid part is 55%. The liquid part is nothing but plasma. Okay, the anticoagulant substance present in the blood. That's why it is a plasma is also having anticoagulant substance. So that is nothing but the heparin. Heparin is the anticoagulant substance present in our body. That's why blood is not clot. Suppose if it is absence, what happens? It will lead to clotting. At the same time, a mismatching of blood. Already when any one of the blood transfusions. So corresponding blood group only you have to transfuse. Otherwise, what will happen? It will lead to clumps. As a result, lead to death. Okay. When you are going to study the blood transfusion, before that mismatch That things we have to study. What are the types of blood groups? 4 types of blood groups there, isn't it? What about the antigen? Same antigen is there, but different antibodies is there. Suppose if it's a blood group A, the antigen is A. Antibody B. When you are suffering any accident persons, blood group is A, we have to provide only A blood group only. Or suppose we are alternatively used, what will happen? Wrongly we are introduced, what will happen? Suppose the antigen A and the antibody A also mixed together, as a result of formation of what is that? Clumps, a blood clotting mechanism happened. So as a result, lead to death. We will see that things in when you're studying the hematological studies. Okay, go to the next one. So antibodies are third line of defense. They are proteinous substances present in the blood plasma. These are complex glycoprotein molecules produced by the B lymphocyte. Note it down, B lymphocytes, B denoted as bone marrow. This response to an antigen, it is also called as agglutinin. When it reacts with the antibody, antigen reaction, what happened? It is nothing but the clumps or clot like that. That condition is called as agglutinin. The process is called as agglutination. Sorry. Okay, that is a thing. See here, this is the structure. The normal immunoglobulin, otherwise called as antibody structures, look like a letter Y-shaped in nature. Once again, repeat for you, Y-shaped. Why Y-shaped? It's look like a Y-shape. How it will be maintained their structure? Will you please, anybody? How they are maintain their Y-shaped structures? Ajay, Dhanalakshmi, Ritika.
So it's because of the heavy chains and light chains, sir.
Heavy chain and right chain is okay, but there is a bond in between, isn't it? What is that?
Yes.
See here, that's simple. Yeah, that is called a sulfhydryl bond. That is very, very much. That's why it's look like a wine shaped. Okay. So this is the two things. Is that see that outermost chain is called as light chain. This is the chain. It's called as light chain. The innermost chain is called as heavy chain, because see the structure is also a long one, isn't it? Compared to the peripheral, isn't it? The light chain always two in nature. Once again, repeat for you. The light chain always it has two types only. One is called as kappa or lambda. It is denoted as K or L light chain. These two chain, the light chain is there, no? Here is the light chain is one side. Here also the another one is there, isn't it? These two are the outermost chain is called as light chain. Okay, they are having two types. One is kappa chain or lambda chain. But take an example of heavy chain. See that heavy chain is the inner molecules, inner molecules. Okay, the inner molecules, see that how many changes is there? See that have been reported 5 types of heavy chain is reported here. Immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin D, immunoglobulin E. See, these are the types. These are all our heavy chain. So in between there is a bond is there, that is called a sulfhydryl bond. So just to go to the next slide, you can see clearly, isn't it? Okay, see the structure. So you can see the exact correct way. See that this is the variable chain. So it is a reversible manner. It's outside or inside. Okay, see the next one we will see. See, this is the correct one. Okay, see here, this is VL, isn't it? And CL. So VL is CL. What is that? Light chain, a variable region. This is also another bond. Okay, inner side, the heavy chain is there. See that this is called as heavy chain. Okay, the heavy chain, it has a two component connected with the disulfide bond. This is called as disulfide bond. Okay, so inner side there is a heavy chain, and also outside there is a light chain. Okay, that is it has a amino acids and amino end is there, because it's made up of protein molecule. That's why. So this region is it is called as antigen binding region. This region is called as antigen binding region. See here, So the both the side, they are having antigen binding sites. Each antibody, how many antigen binds? No, any one of the immunoglobulin G or A, like that, See that two antibodies bind with the each two antigen bind with the each antibody. This is antibody, or otherwise called immunoglobulin. The antigen binding site is the one side. So this is called as Fab region. This is called as Fab region. This is variable region. This one is called as constant region. This is called as constant. C denoted as constant. V denoted as variable. Variable light chain and constant light chain. Variable heavy chain and constant heavy chain 1 and constant heavy chain 2, constant heavy chain 3. See that here the amino end, here the carboxyl end. Because each one it's considered as which one a single polypeptide chain, otherwise called as globulin protein. Once again repeat for you. The immunoglobulin is Y-shaped. It has a light chain and a heavy chain. The light chain also divided into two parts. A variable region. This region is called as variable. VL denotes as variable region and CL denotes as constant region. Likewise, the heavy chain also divided. So variable heavy chain and constant heavy chain 1, constant heavy chain 2, constant heavy chain 3. So how many constant chain present in the which one heavy chain? So there are 3. See, CH1, CH2, CH3 is a constant region. Only one variable region is there, one side. So another side is also same. 2 variable region. How many constant region? There are 6 constant region is there. Connected with the disulfide bond. See here, the disulfide bond is here. The heavy chain, light chain also connected with the disulfide bond. And also having this region is called as Hinch region. Once again, repeat for you. Light chain, heavy chain connected with the region is called as Hinch region. This is called as Hinch region, this area. Okay, there is a flexible manner. This is folded structure already you are seeing, isn't it? The structure is called as Hinch region. Okay, collectively they are all are called as what is that? Antibody. Okay, so this is the structure of antibody. See one by one what are the things is there. See here. See, each antibody made up of 4 polypeptide chain. It has a 4 polypeptide chain. 2 long chain is called as heavy chain. 2 short chain is called as what is that? Light chain. Each of molecular weight, this is called as a heavy chain molecular weight. It denoted as a Dalton as a unit. Heavy chain 50,000 Dalton. Short chain is called as light chain. Each of molecular weight 25,000 Dalton. Hence, it is represented to heavy chain 2, light chain 2. Already you saw it in the diagram, isn't it? Or slide. These 2 chains held together by disulfide bond in shape of Y molecule. That is the relationship. How it will be standing with the help of the bond is called as disulfide bond. How many heavy chain is there? 2 heavy chain is there. How many variable region is there? Each one 3 variable is there. Okay. See, light chain 1 variable, 1 constant range. How many molecular weight is there? Light chain is 25,000 and heavy chain is 50,000 Dalton. Total for polypeptide chain. So you note it down. So, and 2 top tips of the this bind with the antigen, large and complex foreign molecules, mainly protein that activate the specific immunity like lock and key. So each and every protein molecules such as the antibody, they will not bind with any one of the antigen. They exactly bind with the which one a specific antibody and specific antigen only. Then only the antigen antibody complex will be formed. Otherwise, it will not form. Just like a lock and key model, when you are going to study the enzymology, the specific substrate bind with the specific enzyme. As a result, you will get it an enzyme substrate complex, isn't it? Likewise, the antigen antibody is also specifically binded. That is very, very important part. Okay, go to the next one. The heavy chain is also called as H chain, already we discussed, whereas the light chain is called as L chain, which may be either lambda or kappa. Lambda denoted as L, kappa denoted as K. Already we discussed lambda as well as the kappa. See, this is the variable region, isn't it? But in this category, see that this area will be differ according to their mu. That is also called as M, immunoglobulin M. This is called as gamma, G. What is that? Alpha, A, delta, D, or epsilon, E. Likewise, according to the heavy chain, constant heavy chain, they may be classified into fighters. We will discuss what are the types of immunoglobulins. Okay, you just imagine game D, game GAME-D, like that immunoglobulin G, and like that you have to identify. Okay, so okay, we will see. There is a variable portion for binding to antigen through electrostatic interaction and the constant portion that determine its adherence and diffusivity. Variable region is also called as B regions. What is that? B region. Antigen binding, fragment and fab. Fragment and fab. That is region is called as what is a variable region is also called as V regions. Antigen binding site is called as a fab region. Antigen binding region is called as fragment region, otherwise called as fab region. Okay, got it. So this is already we discussed, isn't it? See that light chain and heavy chain. Okay, so here this is the site of antigen binding site. This area as well as this one, antigen binding site. Okay, the opposite side, this is a carboxylic end. See that carboxylic end, and opposite side there is a amino end is there. This is light chain, this is heavy chain. How many variable region is there? See here, see these are the variable regions. So 3 variable region is there. Okay, so that is called as FC region. These are the FC region. This is called as FAB region, FAB, antigen binding region, FAB region. Okay, got it? With the help of the both molecules connected with the hinge region, just like a folding of molecule with the help of the disulfide bond also. Okay, this is called as antibody structures or immunoglobulin structures. They may ask the question so many times, without immunoglobulin, there is no immunology, isn't it? That's why. Okay, see that. How many types of immunoglobulin? They may be divided in 5 types. Immunoglobulin G, you must note it on here. How many percentage of immunoglobulin present in plasma? That should put a chart and take a notes. Immunoglobulin G, 75%. Immunoglobulin A, 10 to 15%. Note it down. This is very, very important. So many times they were asked the questions. Take a short notes. Okay. And then immunoglobulin M, 5 to 10%. Immunoglobulin D, more than 1%. Immunoglobulin E, 0.1, more than 0.1. Suppose if it is the immunoglobulin E is increased, what happened? You are in allergic condition. Already we discussed. When the immunoglobulin structure or immunoglobulin, the percentage will be high in your blood plasma. What will happen? It will lead to what is a hypersensitivity. Okay, so we'll discuss one by one. See here, and immunoglobulin G, 75%. What is the characteristics and functions found in blood, lymph, and intestine? Active against bacteria, is toxin and viruses. Enhance phagocytosis. What is meant by phagocytosis? Is nothing but the cell eating process. We will discuss last. Already I told you. Okay, otherwise called a cell eating process. Simply I tell you, okay, any one of the foreign particle in bad, what will happen automatically? The suicidal bag, lysozyme enter, react with the foreign particle and produce that enzyme is called as lysozyme. Lysozyme activate upon the invading particle and completely lysis. Lysis, after that, what will happen? The complex molecules converted into simple molecules. Finally, the waste will be eliminated through outside. Okay, so that is a process. You must note it in here. Immunoglobulin G, 75%, where it present blood, lymph, and intestines. What is the main activity? It activity against bacteria and virus. Okay, and also it is responsible for the phagocytosis. Crassus placenta, and it is act in a second response. Note it down. This is called a second response. Okay, and immunoglobulin A. Immunoglobulin A. How many percentage you know? 10 to 15%. Here, saliva. Your saliva has which one you know? Immunoglobulin A, because antibacterial activity. How it will be act as antibacterial activity? Because take an examples of dog. It has some wound, you know. Only they use saliva as a medicine because saliva contains more amount of which one you know? Immunoglobulin A. Tears, lacrimal gland, and bronchioles, GI, and what is that? Gamma, prostate, and also vaginal secretion. These are the which one you know? Immunoglobulin A. Provide local protection on exposed mucous membrane surface and potent antiviral activity. Same here, but it may local. And also prevent the absorption of antigen from food and protect against respiratory and gastrointestinal and also GU, gastrourinary infections. Here, gastrointestinal. These are the another category. Immunoglobulin G, immunoglobulin E, percentage where it found. Okay, just what are the main functions of the antibodies. So next one is immunoglobulin M. It is 5 to 10%. Level decreases during stress. Suppose when you are having the stress, when you are under the stress, the immunoglobulin M level is come down. Found in blood and lymph, similar to that of immunoglobulin G. The first antibody produced with the primary immune response. Which one is the first antibody response? You know, produced, which you know, immunoglobulin M. Note it down. This is very, very important one. High concentration yearly in infection decrease within about a week. Suppose any one of the diseases, what happened? You know, it may be high in nature. Immunoglobulin M is during the any other infection, the immunoglobulin M is very high in nature. After that, it will come to the normal position. Immunoglobulin D, less than 1%. Sorry, more than 1%. Unknown function found in blood and lymph. Immunoglobulin D, we do not know the function exactly what happened inside the body, what is it doing inside. The things are completely lack. Immunoglobulin D, already I told you, more than 0.1%. Suppose if it is increased 0.3, 0.5, you know, you are having some difficulties in your blood. Because found in mast cell and also basophils. Mast cell is very, very important. So many times they were asked, immunoglobulin E is secreted, anti-allergic substances secreted by the mast cell only and also basophils. When it in, how it will be? When it, where? Okay, involved in the immediate hypersensitivity response. You know about some of the person, they took dry fish, even brinjal. Okay, sometimes tramarindus. Okay, when you're taking, what happened? You will get the small patch like in your skin, readiness. Okay, the symptom you may observe. That is due to the allergy. Due to the allergy, there may be a dust allergy or sometimes food allergy also is happen. What happen automatically? The mast cell secreted immunoglobulin E. Okay, that person just go and observe in the blood hematological analysis. You can see the more than that of the immunoglobulin E. This is maybe up to the level of what is that? 0.5%. 0.5%. It lead to hypersensitivity response. Okay, this is also very, very important. Okay. See, these are the structures. See, the immunoglobulin M is a pentamer. Sometimes they may ask the question according to their structural aspects. This is a pentamer. See here, how many Y-shaped you know? So this is also one Y-shaped, another 2, 3, 4, 5. That's why the 5 antibodies, this is a one. Okay, this is a single. Likewise, 5 antibodies united with the which one? The center. Okay, that's why it is called as immunoglobulin M is a pentamer The M denoted as mu. M denoted as mu. Okay, they are all united with the help of bond. How many antigen binding bond here? So many times they were asked the question because it has a 5, isn't it? Suppose if it is single, there is a 2 antigen binding site is there. We discussed it. So 5 antibody is there. That's why here given as a 10. Okay. And one more is that what about the molecular weight among these 5? Which one is the higher molecular weight? You know, immunoglobulin A is M is the higher molecule. 900,000 Dalton. Note it down. This is very, very important. Highly specific. Pentameric nature. Mu denotes M. Number of antigen binding site is high compared to other components. 10. Molecular weights also, how much? 9,000. The percentage of total body in 6%, total serum. Okay, serum already you know after the blood clotting, you will get the yellow color fluid is called as serum. Okay, so 6% will be. Next one is immunoglobulin G. It is a monomer. Immunoglobulin E is also monomer. Immunoglobulin D is also monomer. And security immunoglobulin A is a dimer because 2 antibody united with the help of the what is a bond. Okay, the immunoglobulin M we saw it. Immunoglobulin G is a monomer. See here, structure already a heavy chain, light chain is connected to the disulfide bond. The G denoted as gamma. Antigen binding site 2 only. Already see it here. Here is also 1 and here is also a 1. So there are 2 is there. But here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That's why here 10. The molecular weight will be 150,000 only. But most of the cases, immunoglobulin Mg will be high concentration. How many percentage? This is also very, very important. 80% of the blood serum contain, which one you know? Immunoglobulin G only. Okay, this is very, very important. Next one is, see that secretory immunoglobulin A dimer. Dimer means 2 antibody united together or 2 immunoglobulin united together. It is also called as another one of the specialized structure present in it that is called a secretory component. Sometimes they may ask the question, which one of the following is a secretory component immunoglobulins? Already they have given the heading also same, secretory immunoglobulin A. Okay, the A denoted as alpha. Here the 2 antibodies there, so antigen binding site is also 4 because here 1, 2, 3 and 4. The molecular weight, see here 385,000. How many percentage? 13%. Okay, and immunoglobulin E. So this is also similar. It's also monomer. Already we discussed it. The symbol is epsilon. E denoted as epsilon. So the immunoglobulin is also a 2. Here also 2 because it's a monomer. Monomer it consists of 2 only. Pentamer is multiply into 2. Dimer into multiply into 2 again. Okay, so you'll get 2. What about the molecular weight? 200,000. The percentage already we discussed here. 0.002% present in the serum. Very, 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 very less in nature. Immunoglobulin E is very, very less in nature. They may ask the question, which one is the less component? Which one is the highest amount present of serum? Which one is the higher molecular weight? Which one is a pentameric nature? Which one is the dimeric nature? Which one which of the following is a monomeric nature? Like that they may ask the questions. The last and the least immunoglobulin D is also a monomer. The D denotes a delta. Monomer means about the 2 antigen binding site is there. And for many molecular weight 180,000. And percentage will be very least compared to which one other 3, isn't it? This one is very least, but here the 1% is there. Compared to this one, which one is the lowest molecular weight? Just imagine lowest molecular immunoglobulin G is a lower molecular weight. Which one is the higher molecular weight? Which one is the immunoglobulin M? Okay, in order to binding site also. So many times they were asked the question according to the structure. according to the number of antigen binding site, according to the molecular weight, according to the number of percentage present in the blood serum. That things, just to note it down. This is very, 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 very important. Don't forget it. At the time I have examination, without this portion, okay, the question is not there. So be careful and study well. Okay, see, and percentage of total antibody in serum, already we discussed this part. Okay, and crosses placenta. Okay, just take an example. The first one, it will not cross the placenta, except immunoglobulin G. It will not pass the placenta, umbilical cord. So the immunoglobulin G only cross the placenta. That is very, very important. Compared to other four, they will not cross the placenta. And also fixes complement. Immunoglobulin M and G is having the fixes complement, and other three is not. And FP binding to, here there is no, this is phagocyte, this one is not there, and the mast cell and also basophil is there. This one is called as immunoglobulin E. The functions already we discussed. Main antibody of primary response. Okay, best at fixing complement, the monomer form of immunoglobulin M serves as the B cell receptor. This is the important functions. Okay, and main blood antibody of secondary response, neutralize the toxin. This is also another. Okay, opsonizations. Already you know what about antigen antibody. When it react, what happen? You know, they will do the neutralization of the toxin, and another one is called as opsonization. These are the two processes done by the antigen antibody reaction after the entry. Okay, see that secrete into the mucus, tears, saliva, colostrum. Colostrum is very, very important. We will know what is by colostrum. The first milk of mother, isn't it? It's a pale yellow color, which immunoglobulin is highest. See that one? Immunoglobulin A is the highest concentration in the, which one you know? And then immunoglobulin, antibody of allergy and also anti-parasitic activity. Immunoglobulin E, antibody of allergy responsibility of the molecules. And last one is called as beta cell receptor. So these are all are the functions and also tabulation given by the scientists regarding their activity and then function, how many percentage, structures, and also their name. Okay, and also Roman letter. See that immunoglobulin G, gamma, immunoglobulin M, mu. M denotes mu, G denotes gamma, A denotes alpha, D denotes delta, E denotes epsilon. Structure will be same, but these two are different. Already we discussed. This is called a secretory. This one is called a pentameric nature. Okay, so already we saw it, these things very clearly. Okay, so first of all, you should know drugs expert, how they will be divided. That things is also no need. Already we discussed these things. Okay, see, and then we will study one by one. Already we discussed immunoglobulin G, classes, properties of immunoglobulin G, molecular weight 150,000 Dalton, chain type, molecular weight H chain, heavy chain type, molecular weight gamma, already G denoted gamma, 53,000 Dalton. Serum concentration 10 to 16 mg per ml. Serum per ml 16 mg percent. Percent of total immunoglobulin 75%. Immunoglobulin G will be highest concentration. Already we discussed. And glycosylations, it's one of the processes. Glycosylations by weight 3%. Distribution intra and extravascular function secondary response. You have to find out here molecular immunoglobulin, molecular weight, percentage of serum, and percentage total immunoglobulin in blood, glycosylation, where is the distribution, what is the main functions. You have to put the tabulation and fill it properly. You will get the here very, very important things, you know, these two things is very, very important. Serum concentration and percent of total immunoglobulin in blood, 75%. And what is the response? What is the functions? Mainly secondary responses, very, very important. And then see that immunoglobulin G structures. See here, sometimes they may ask the question, heavy chain contain how many residues? Okay, 450 residues also present in it. The light chain, 212. Okay, 212. Already we discussed these things all are already, So this is called as carbohydrate. Disulfide bond results. Heavy chain, light chain. CH1, CH2, CH3. These are all are, which are, heavy chains. Okay, these things. What are the next one? Immunoglobulin M. It is also nothing but pentameric nature. 5. Already in our molecular weight, already we discussed 900,000 Dalton. Heavy chain type, molecular weight mu 65,000 Dalton. Okay. And serum concentration 0.5 to 2 mg per ml. Percentage of total immunoglobulin 10% only. Glycosylations 12%. Distribution mostly intravascular. There extravascular and intravascular immunoglobulin G, but in this category only intravascular only. Function, it is also a primary response. Okay, already we saw it. This is the structure of the which one, immunoglobulin M. See here, so J chain, this one is called a J chain, joining chain. Okay, 1 2 3 4 5 united together. Okay, the extra domain CH4 extra domain is also present in it. And J chain, pentamer 19S subunit. 19S means it's also made up of protein molecules. Okay, so these are the very, very important things. See that. And then immunoglobulin A classes, already we discussed, molecular weight to 320,000 Dalton. It is also called a secretory immunoglobulin. Immunoglobulin A is a secretory immunoglobulin. Heavy chain type, molecular weight is alpha. A denoted as alpha. 55,000 Dalton. Serum concentration 1 to 4 mg per ml. Before 1 to 2 mg. Here 1 to 4 mg. Percentage of total immunoglobulin 15%. Above 10%. Glycolysis by weight 10%. Distribution, it's also intravascular and secretion. What they are going to secrete? So many things they may secrete. What are they? We will discuss. Functions, protect mucous membrane. Secretion means what is that? Lacrimal secretion, mucous membrane secretion. That is the things. Okay, got it. Okay. So function is protect mucous membrane. Mucous membrane where it already we studied, nasal cavity, urinary tract, and also small intestine and large intestines. This is very, very important. See the structures. So this region is called as antiband Fab region, antigen binding site, this region. Likewise here also both. The joining chain, here the one more is that joining chain is here. Okay, this is the secretory protein. This is called as a secretory protein. So the presence of secretory protein, the immunoglobulin A is also called as a secretory immunoglobulins. Okay, so this is the structure of immunoglobulin. It is a dimer, already know. But dimer means two immunoglobulin united together with the help of the joining chain. Okay, so sometimes they may ask the question. That's why. Immunoglobulin D classes, a properties of immunoglobulin D at the molecular weight at 180,000 Dalton and heavy chain type. All are heavy chain types. Molecular weight delta 70,000 Dalton. Serum concentration 0 to 0.4 mg per ml. Percentage of total immunoglobulin 0.2%. Glycosylation 13%. Distribution lymphocyte surface. Okay, lymphocyte surface. For function unknown. Already we discussed it. Which one of the following function is unknown? Like that they may ask the question in immunoglobulin D only. Okay, so this is important. And then last, see the structures. These are all our monomers. Already we discussed immunoglobulin D, immunoglobulin E, immunoglobulin G. This is called as dimer. This is the pentamer nature. Okay, to the next. Immunoglobulin E is the last. I think D is also that. Okay. Immunoglobulin E is a last. The properties of immunoglobulin E, the molecular weight is 200,000 Dalton. Heavy chain type. Epsilon, E denoted as epsilon. Already told you. 73,000 Dalton. Serum concentration, see here. 10 to 400 nanogram per ml. The percentage of total immunoglobulin 0.02 percentage. Okay. Glycosylation by to 12%. Distribution, let's just imagine, very important. Basophil and mast cell in the saliva and nasal secretions. The immunoglobulin E is anti-allergic substance already in our life. Protect against the parasites and also anti-allergic substances. Okay, so many times they were asked the question, immunoglobulin percentage. See that 0.02 percentage. Where it's secreted, mast cells. What is the main function of this one? This one. Okay, so very important. Don't forget these things. Very important. See that the FC receptor, mast cells. Okay, how it will be? The immunoglobulin E is here. It's bind in the FC receptor. That is called as Fab regions. Okay, receptor cell is here. See the binding of antibody. Here this is called as antigen binding site. That is called as Fab region. As a result, what happened now? The mast cells are completely. This is a diagrammatical representation. Mast cell, how it will be bind. Okay, like that they showed, isn't it? Okay. So once again, this is the, what is that? Mast cell. Okay, this is the antibody E. The antibody bind with the mast cell with the help of the FC receptor. Receptor only receive the immunoglobulin E molecules. So easily bind with the receptor molecule as well as the mast cells. Okay. And suppose if it is two receptor present in here, the one receptor is there, here also the two. When the antigen is bind here, automatically what happen? You know, see here how it will be the antigen binding. As a result, it may be destroyed. See that mast cell or as basophil FC receptor for immunoglobulin type of hypersensitivity. As a result, release the histamine and histamine-like substances, eosinophilic and hemostatic factors. These are all anaphylactic reaction, urticaria, and bronchospasm. These are the things released by the mast cells, isn't it? Histamine, already know about the histamine secretion is also one of the substances that is induce the allergic, isn't it? It was how it will be rectified, you know, when you are taking the antihistamine substances, that is called as what is that, citrusin or avil, like that, you know, when you are taking the tablets, it can completely control it, and it is not a permanent, but it is controlled. Okay, this is very important. As a doctors, you should know very important thing, because this is so many persons affected by these diseases. Anti-allergy substance, you should note it down. Okay, it's known. Okay, known one. See, antibody functions, effect, or mechanism. I already told you, when it bind with the antigen and antibody. Okay, when it corresponding antigen and antibody react with the within, what will happen? You know, the functions of antigen antibody reaction. What will happen when it bind with the corresponding antigen antibody? Antigen antibody function is happen. Antigen antibody complexes happen. After that, what will happen? They undergo a further effect of mechanism that is called as neutralization or agglutination. These are the two steps only. What is my neutralization? How it will be antigen antibody neutralization takes place within the body after the foreign particle enter. Some antibodies functions as antitoxin and neutralize the toxin produced by the pathogen by pathogen foreign chemicals. How it will be when in the antibody enter, what happen? You know, the toxic substance produced by the which one? You know, pathogen, virus, bacteria, protozoan, whatever it may be. Suppose you take an example. Here the pathogen is there, no? 10 million pathogen present in your body. Just imagine. Okay, the antibody is also 10 million. What happened? And both are equal, isn't it? After that, equalization, that is called as neutralization. It completely reduce a toxic effect. Suppose 10 million pathogen is there, 20 million antibody is there. What will happen? You know, the 10 million extra antibody present in your body, so completely neutralize the 10 million pathogens. Okay, that is also another things. And the agglutinations, it is already we discussed. Antibodies called agglutinin causes immobilization and climbing of antigen, otherwise called as precipitation, and antigen containing pathogens. So another one is called as agglutination, is another process which occurs when the antibody, what is that, combine with the antigen. What happened, the precipitations formation takes place. As a result, what happened, they will not move here and there. Which one? The foreign particle antigen, they will not move. That condition is called as immobilizations. Okay, this is the another category. These are the main functions of the antigen antibody reactions. Finally, it will be neutralize the, what is that, pathogenic effect or toxic substances. Okay, see here. And opsonization is another one. Occurrence, antibodies called as opsonins. Antibodies are called as opsonin. Antigens are called as, what is that? Antibodies are called as agglutinin. Antigens are called as precipitations. These reactions all happens. Okay, see that, opsonin or adherence. Antibody is called as opsonin. Immunoglobulin G attach themselves to the surface of the antigen containing cells, antigen presenting cells here, to be recognized by phagocytosis. Already we discussed them. When the antigen present in the cell, the antibody move on that along, move on that side. What happens? Corresponding antigen find it automatically. They may come in contact with the antigen, isn't it? Already we discussed corresponding lock and key model like that. It may combine with that. As a result, what happened? The phagocytosis will be possible. As a result, a foreign particle completely denatured. Okay, and then a fourth one is called as complement mediated cell lysis. Complement mediated cells. It means the two cells between, there is another cell is also, what is that, mediator reaction carried out by the another cell. The antigens containing cells are perforated by enzymes produced with the help of the lysine antibodies, example immunoglobulin N, immunoglobulin G, and the cytotoxic T cells. Okay, in between immunoglobulin M and G and then antigen, there is a T cell is also present. That's why it is called as complement mediated cell lysis. Okay, without T cell, this is not, these two antibodies not activated by this methodology. Okay, that is the reason. So here the three combination is very, very essential. Antigen, immunoglobulin, antibody, and also T cell is also very, very important. That's why they have told us a complement. This one is called, which one is complement? The T cell is a complement for this reaction, antigen antibody reactions. Okay, as a result, lysis takes place. So the pathogenicity as well as the toxic effect will be reduced. That is the reason. Phagocytosis. This is very, very, very important. Okay, they lyse the immobilized clump pathogen. They lyse the immobilized clump pathogen or engulfed by phagocytosis. Phagocytosis is nothing but the one of the physiological process which occurs within the cells. When the foreign particle enter, already I told you so many times. Okay, again I repeat for you. When a foreign particle enter, what happen? Automatically, what will happen? It receive the receptor, receive the foreign particle inside the cell. When it enter inside, what happen? The phagocytosis started. So the message reached to the lysosome. Automatically, lysosome bind with the foreign particle or pathogens. Okay, as a result, automatically it secreted so many components. That is called as hydrolytic enzyme. Lysozyme is an enzyme secreted by the lysosome. So as a result, the pathogen completely denature, and also what are that is called as cell eating. Suicidal bag. Lysosome it also act as a suicidal bag. It kill itself. Okay, not only that, and also pathogen too. Okay, that is also very, very much. See that this is the process, phagocytosis. Okay, see here, this is a leukocytes absorb the bacteria. See that this is considered as a leukocytes. Okay, leukocytes is nothing but the WBC. Okay, here the structure given is a neutrophil structure, I think. Okay, this is the foreign particle. It's absorbed. Leukocytes ingest bacteria, completely ingest the bacteria with the help of the lysosome enzymes. Okay, so leukocyte expand form, ingesting large number of bacteria. Suppose a large number of bacteria enter inside, it digests with the help of the enzymes secreted by the lysosome. As a result, what happens? It absorbs the nutrition from the digested bacteria. So as a result, what happens? The cell size will be, what is that? Large nature. Okay, this is the one step. And again, what happened? One more is that, and lyses, and also white blood cells lyse releasing cytokinin chemical, which cause a local inflammatory reaction. This is another step. After this one, to undergo the lysis process, then it will release the cytotoxic substances. Okay, and then cytokinin attract the new leukocytes to fight bacteria. This is the phagocytes occurs in a single cell only. Okay, phagocyte is nothing but the physiological process which occurs in the leukocyte, particularly neutrophils. It receives the molecules and absorb it, digest it, ingested, and also absorb all the nutritive substance from the bacteria, and also it enhance, or it is a ingested large number of bacteria, and also it what happened, lyse itself produced which one? Cytokinin. So we come to the conclusion, after the enlargement of the cells, what happened? It produced one of the cell is called as which one? A cytokinin. So we come to the conclusion, cytokinin is very, very important for the killing of microorganisms, particularly fight against many diseases of the bacteria. Cytokinin secreted by the white blood cells, because already told you this is called as neutrophil. So white blood cells lyses releasing cytokinins. Okay, and also responsible for the another axon, second axon. This one is, we may consider as a primary axon. This one is called a secondary axon. Okay, this is one process. See the another process. See the another process. This is considered a cell. The center there is a nucleus is there. See the microbes received by the receptor. This is considered a red color. It denoted as a what is that? What is that? Microbes. The receptor is that. So the receptor receives the microbes. This is the step first one, the binding and absorption. After that, it may be enter into the phagosome formation. Phagosome is nothing but the some, lysosome. Act upon it. So act upon it. This is called as lysosome. See that lysosome act upon it. It's here the second stage. Okay. After that, what happens? It secretes so many. What is that? Enzymes. There are 60 enzymes have been reported by this lysosome. So after that, see that phagosome and the lysosome. The phagosome as well as the lysosome both are involved in the, what is that? Ingestion of the microorganisms, particularly as the bacteria. After that, what will happen? Completely digested. Okay, see that digestion takes place. Fourth one. Then last, what are the absorbed material we have to take? All are absorbed and then release the microbial product. Release the microbial product. What are the waste material is present in it? It may be eliminated through the opposite side. It is enter one side and also release the opposite side. So this is the very, very important steps happen in the body. Okay, note it down. It's a very, very important things. See, so today this one is finish it. Okay, today the fifth human welfare is finished. So go through thoroughly, isn't it? Then only it will be better for your understanding. Okay, I ask you questions. What is by phagocytosis?
So,
Yeah.
so phagocytosis, it's the engulfing of any external bacteria or virus by the antibody.
Okay, otherwise called as cell eating. Okay, otherwise
Yes,
called
sir.
as cell eating or cell digestion process. What are the two components essential for the digestion of the bacteria? Lysosome and phagosome, isn't it? Lysosome, how many enzymes they may be secreted? Particular lysozyme is the main enzyme. Okay. Okay. Which one is the dimer of immunoglobulin? Which one of the following is the dimer immunoglobulin? Immunoglobulin?
So,
IgA.
Yeah, good, super. Immunoglobulin A. Okay. Which one is the lowest molecular weight?
so IgE.
Very good. Which one is the higher molecular weight?
So IgG.
Higher molecular weight. Which one is the higher molecular weight? Ajay?
So molecular weight, sir, sorry, sir. I thought percentage, sir. So
Okay.
molecular weight IgM, sir.
Yeah, because why it is called as why it has a high molecular weight?
So pentamer structure, sir.
Pentamer. 5 antibodies united together with the help of the bond, isn't it? That is the reason. Lowest one means you just note it down. Which one is the lowest one? Immunoglobulin G only. What is my colostrum?
Sir, thick yellowish fluid present in the mother's milk, sir.
No, okay. First milk. You have to tell us the first mother milk. Okay, that is which immunoglobulin is high in cholesterol.
Sir, IgA.
Good, super, very good, exactly correct. Okay, so good. So, so any other question regarding this portion, Paul?
No, sir.
Okay. Do you have any idea? Do you have any clarification in this area, please? Okay, we'll pass on to the next one. Just I give you an introduction only. Okay. So next we'll pass on to the portion, part 6. Okay, just I give you an introduction and then we'll finish. Okay. So we'll pass on to the next one is part 6, human welfare. Okay. All the best for the future doctor. Here we have to discuss type of acute immunity. What are the type of acute immunity? They divide into 2: active immunity. Another one is called as natural active immunity, and then passive artificial active immunity, natural and artificial. What are the cells comes under this heading? Okay, and what is by vaccination, attenuated vaccination, how it will be act on passive immunity? What is meaning of passive immunity? Okay, and what is my colostrum? Okay, colostrum, listen here, this is very, very important already. Dhanalakshmi told, isn't it? Good. Colostrum is rich in immunoglobulin A. So many times they were asked the questions. Okay, so this is also very, very important. Immunoglobulin A is also one of the dimer condition. Note it down. Don't forget. So this is our monomer. Artificial passive immunity, and also how the vaccination are prepared. Okay, from the horse, how it will be neutralized, and primary and secondary immune response. Okay, and how it will be vaccination are prepared. Who is the father of vaccination? Who discovered first? Okay, that things is also we have to discuss. And what are the 2 types of vaccination is there? Okay, life attenuated, killed, biosynthetic vaccination, toxoid vaccination, and that things also we are going to discuss in the next classes. Okay, so then first generation vaccination, second generation vaccination, and also third generation, fourth generation, fifth generation wise until the 5th generation. How many types of generation is there? See here. We are all are in the which generation? You have to note it. Just go and check it in your which one you know. Google. Okay, search it. We are all are in which generation now we are living. Okay, that things how many times this is also very, very important. So many times they were asked the question vaccination program for the 0 to 5 years baby. At the birth of time, which one they are providing? Which one is the first dosage? PCG only. See that PCG. Okay, and also oral polio. So oral polio vaccine is also hepatitis B1. These are all our first one at birth. 1.5 months, 2.5 months, 3.5 months, 6 months, 9 months, 15 to 18, 4 to 5 years. Likewise, you have to take up to 5 years. First standard. Take after that. No need, I think. Okay, because these are the things. Immune disorder is also that. And what are the difficulties it happen inside? Okay, so keep it in your mind. Here, you last class, what you have to know? You know about what is my immunology, immunoglobulin, types of immunoglobulins, and then structures, molecular weight. Okay, that things already we discussed. Okay, thank you so much for your participation. Just to go through it, you will get it idea. All are attending the telegram of the question part.
Yes, sir. Sir, I attended, sir.
Oh yeah. So if it is okay, or any other difficulties you are facing.
Sir, it's okay, sir.
So better, compared to the previous one, isn't it? What about the analysis?
Yes, sir.
What about the analysis? You didn't answer me.
Sir, today I will attend, sir.
Okay, but it's okay this way. Okay, or any other changes if you need.
It's okay, sir.
Okay, how many questions? I think in 11 questions I have given, isn't it? Suppose we want more questions, I think we may
Sir,
give
16 questions you gave, sir.
16. No, today.
Sir,
Yeah.
yesterday, sir, you gave 16 questions.
Okay. Anyhow, so 16 is enough, or 15 is enough, or 20 need more questions? How many minutes you took for this one?
So 5-10 minutes, sir. Less than 5 minutes, sir.
Yeah,
5 minutes, sir.
5 minutes is enough. Okay. Because you have to attend more questions. That is very, very important. Okay, then only it will be practice and also feed in your memory. Okay, so practice only make the man perfect. That is very, very important one. So you have to practice it properly and go through it before, when you are going to study the lesson. Human welfare, I told it. Okay, so you have to when you are starting this lesson, just to go through it. So then only when we are taking the classes, it will be easily better understand. Okay. Pa. Thank you so much for your. We'll see the remaining on the next class. The part 6 we'll see the day after tomorrow. Okay. Bye. Take care.
Thank you, sir.
Thank you, sir.
Thank you, Pa. Thank you. Thank you, ma'am.
Thank you, sir.
Thank you, ma'am. Bye.